Welcome to the deep dive. Today, we're tackling a really fundamental question in biology. How does a single fertilized egg actually become, well, us, a complex human being with all these different cells and organs?
Right. And also, how do those tiny, tiny changes in our genes sometimes lead to really complex diseases?
Exactly. For a long time, the tools we had gave us, pieces of the puzzle, but never the full picture, especially not how it changes over time.
That's a great point. The Human Genome Project was absolutely monumental, right? It gave us the complete DNA blueprint.
Yeah, the whole sequence.
But even with that map, this huge question persisted. How does that genetic code actually orchestrate everything? How does it guide development and drive disease when you factor in where things are happening and when?
It's like having the plans for a skyscraper, but not knowing how the materials get put together on site or in what order.
Precisely. It's about the dynamics, the spatial and temporal context.
And that brings us neatly to our topic for this deep dive, spatiotemporal omics. This sounds complex, but think of it as a revolutionary way to combine omics, mapping genes or proteins, with super precise information about where molecules are and when they're active. So our mission today is to unpack this. How does this technology let us create Google Maps for the human body, revealing things we never knew and answering some really basic questions about life?
Sounds good. It's a fascinating area.
Okay, so let's dig into that foundational challenge a bit more. We remember the central dogma, right? DNA makes RNA, RNA makes protein. And the Human Genome Project gave us that full DNA sequence.
The blueprint.
Right. But the really tricky part is, how does that one genome, present in that first fertilized egg, know how to generate such a huge diversity of cells? Muscle cells, nerve cells, skin cells, all from the same instructions.
That's the core of development, yeah. And beyond that, there's the disease angle. We found loads of genetic variations linked to diseases through big studies like GWS. But the question is, how do those specific variations actually affect, say, your liver or even specific cells within your liver? What's the mechanism in that specific context?
So it's about connecting those tiny molecular sequence details to the functioning of a whole biological system, cell by cell.
Exactly. And to do that properly, we really need to understand 5 key dimensions of how cells organize. First, qualitative, just the sheer variety of cell types.
Okay.
Second, quantitative, how many of each type, their density. Third, and this is crucial for today, space. Their physical arrangement, how they're positioned relative to each other, their neighborhood.
Where they are on the map.
Precisely. Fourth, time. The specific timing of when cells change, type, or state. And fifth, direction. The potential paths a cell might take, its lineage or transformation potential. Getting a handle on all 5 is key.
And that's where older technologies kind of fell short, right? Like bulk sequencing just averaged everything out.
Yeah, you completely lost the cellular differences. Then single-cell sequencing was a huge leap forward. We could see individual cell types.
But you lost the where. You took the cells out of their environment.
Exactly. The spatial context was gone. And traditional imaging, like MRI or histology, showed structure, but didn't give you that deep molecular information for thousands of molecules simultaneously.
Which leads us to
Spatial omics. The whole point is to get that rich omics data, transcriptomics, proteomics, whatever, but keep the spatial information. Know exactly where each molecule was in the tissue.
so you can map the cells, see who's talking to whom, watch the dynamics unfold.
Exactly. It's essential for understanding how tissues are built, how they work, and how diseases mess things up at that local level.
Okay, this makes so much sense. So let's get into the how. How do these spatial omics methods actually work? What are the core ideas that let them pinpoint molecules like this?
Well, the fundamental principle is pretty straightforward conceptually. Detect the molecules you care about, RNA, DNA, proteins, right where they sit in the tissue sample. And broadly, the methods fall into 2 main camps, imaging-based and sequencing-based.
Okay, let's tackle imaging first. How do you see specific molecules in their native location?
Think of it like using tiny, highly specific molecular flashlights. These are usually nucleic acid probes, often tagged with something fluorescent. You design these probes to bind only to your target molecules, say a specific RNA sequence.
Like a key fitting into a lock.
Exactly. Once they bind, you use a powerful microscope to take pictures, and the bright fluorescent spots show you exactly where your target molecules are. The earliest technique like this was single molecule FESH, or CEFM-FESH. It could literally visualize individual RNA molecules.
Wow, single molecules. But I guess the challenge was seeing lots of different types of molecules at once.
Precisely. You hit what we call the color wall. Microscopes can only distinguish a limited number of different fluorescent colors simultaneously, maybe 3 or 4 reliably. So initially, you could only look at a handful of different RNA types in the same experiment.
So how did they get around that? If you only have a few colors, how do you end up identifying thousands of different genes?
This is where it gets really clever. They developed iterative methods. Instead of needing a unique color for every single gene target, they used combinatorial labeling and sequential imaging. Think of it like creating a barcode for each RNA molecule using just a few colors over multiple rounds.
Okay, walk me through that.
Techniques like Merfish and Seqfish plus do this. In each round, they attach a set of probes, take an image, wash those probes off, and then attach a different set. A specific RNA molecule might light up in, say, round 1 with the red color, not light up in round 2, and light up in round 3 with the green color.
So its barcode is red off green.
Exactly. That unique sequence of signals across the rounds identifies that specific RNA type. By doing many rounds, you can generate unique barcodes for thousands, even tens of thousands of different RNA molecules, like Seqfish Plus does. And Merfish even builds an error correction, making it really robust.
That's incredibly smart. Using time and combinations to beat the color limit. What about actually reading the sequence in situ? Not just detecting its presence, but reading the letters.
Right, that's the next level, in situ sequencing, ISS. Here, the goal is to directly determine the sequence of DNA or RNA molecules while they're still in the tissue. A key innovation here was figuring out how to amplify the signal locally.
Because a single molecule is tiny.
Very tiny. So they often use something called rolling circle amplification, RCA. You get an enzyme to basically make many, many copies of the target sequence in a little ball right there at the original molecule's location. This creates a much brighter signal called an RCP.
A rolling circle product, okay?
Then, using specialized chemistry, often sequencing by ligation, they read the sequence of that amplified product base by base through multiple imaging cycles. Technologies like StarMap improved this significantly, making it more efficient and allowing detection of over 1,000 genes.
But what if the molecules are just packed incredibly tightly together inside the cell? How do you even resolve them? It sounds like molecular gridlock.
That's a major hurdle, molecular crowding, and the solution is, frankly, ingenious. Expansion microscopy.
Expansion, you make the sample bigger.
Exactly. You embed the tissue in a special kind of swellable polymer gel, like the stuff in diapers, actually. Then you chemically trigger the gel to expand, and it swells isotropically, meaning evenly in all directions.
Wow, so it just stretches the tissue out.
It physically magnifies the sample, maybe 4x, 10x, even 20x, while preserving the relative positions of the molecules. This physically separates those crowded molecules, making them much easier to resolve with a microscope. Techniques like XVASH and XSEC combine expansion with FI or sequencing for amazing resolution.
So, summing up imaging methods, incredible detail, down to single molecules sometimes, but what are the downsides?
The main advantage is definitely that high spatial resolution, sometimes subcellular or single molecule, but the trade-offs are usually complexity and time. These methods can be quite labor-intensive, requiring many cycles of chemistry and imaging.
And maybe sensitivity drops off over many cycles.
That can happen, yeah.
Yeah.
And comparing the abundance of different molecules can be tricky because probes might bind with slightly different efficiencies. Plus, scaling up to very large tissue areas can be challenging.
Okay, that makes sense. Let's switch over to the sequencing-based approaches. These sound fundamentally different. How do they capture spatial information if they're relying on sequencing, which usually means taking the molecules out of the tissue?
Right. The key here is to label the molecules with their spatial location before you sequence them. It's like putting a tiny GPS tag or address label on each molecule while it's still in the tissue.
Ah, okay. How do they do that?
The breakthrough was using spatial barcoding on physical carriers. Imagine a glass slide or a chip surface that's pre-printed or coated with millions of tiny spots. Each spot has DNA probes attached, and crucially, all the probes at one specific spot share a unique DNA sequence, the spatial barcode that corresponds to that spot's XY coordinates.
Like a grid of unique zip codes printed on the slide.
Exactly. So you take your thin tissue section, place it onto this special slide. The RNA molecules from the cells in the tissue diffuse down locally and get captured by the probes directly underneath them.
And they pick up that unique barcode from that spot.
Precisely. The cell's RNA gets tagged with the spatial coordinate of its origin. Then, you collect all that tagged RNA, sequence it, and use the barcode sequence to map each RNA molecule back to its original location on the slide.
Clever. And how has the resolution of this approach evolved?
It's improved dramatically. The pioneering technique, spatial transcriptomics, ST, which is now commercialized as Visium, initially used printed spots. about 100 micrometers across. Good for tissue regions, but bigger than single cells.
Okay.
Then came innovations like slide-seq and later HDST, high-definition spatial transcriptomics. Instead of printing spots, they randomly deposited tiny beads, each carrying a unique barcode, onto a surface. They first figured out the location of every single bead, then put the tissue on top.
So the bees capture the RNA.
Right. And because the beads are much smaller, down to 10 micrometers for slide-seq and even 2 micrometers for HDST, you get much higher, potentially single-cell or even subcellular resolution. Visium HD is also now at 2-micron resolution.
2 micrometers, that's tiny. Are there other ways to get these barcodes on?
Yeah, other cool approaches exist. DBT-seq uses microfluidic channels flowing across the tissue in perpendicular directions to deliver different barcode components, creating a unique barcode combination at each intersection point, around 10-micron resolution.
And have they pushed resolution even further?
Oh yes. The latest leap uses nanoarray sequencing chips. Techniques like stereoseq, developed using BGI's DNA nanoball arrays, or Sucscope using Illumina chips, essentially create incredibly dense fields of nanoscopic spots with spatial barcodes. These can achieve resolutions down to 500 nanometers. Truly nanoscale mapping of the transcriptome.
Wow, 500 nanometer. That's incredible detail.
Okay, we've talked a lot about RNA, the transcriptome, but cells are way more complex than just their RNA, right?
Yeah.
What other molecular layers can spatial omics explore? Absolutely critical point. RNA tells you which genes are potentially active, but it doesn't tell the whole story. Spatial omics is now being applied to map DNA itself, spatial genomics, which is huge for studying things like how different genetic mutations are distributed across a tumor.
Seeing the tumor's genetic evolution in space.
Exactly. Then there's spatial epigenomics, mapping things like chromatin accessibility, which parts of the DNA are open and active, using methods like spatial ATAC-seq, or mapping histone modifications with spatial CT and tag. This tells you about the regulatory landscape of the genome, spatially resolved.
So not just the genes, but how they're controlled location by location.
Precisely. And of course, spatial proteomics. Using antibodies tagged with DNA barcodes, like in Codex or Immunosaber, you can map the location and abundance of dozens or even hundreds of different proteins in the same tissue section. There are also methods integrating imaging and mass spectrometry for deep protein analysis in single cells.
What about translation, seeing proteins actually being made?
Yep, spatial transatomics methods like RiboMap can map ribosome positions, giving insights into protein synthesis within the cell's architecture. And finally, spatial metabolomics, often using mass spectrometry imaging, MSI, maps the distribution of small molecules, the metabolites, linking metabolic activity directly to tissue location and function.
So you can map almost any key molecular layer now spatially. What about putting them all together? Can you measure RNA, proteins, and maybe epigenetic state all from the same spot?
That's the ultimate goal, right? Spatial multi-omics. And yes, techniques are emerging to do just that. CI-seq, originally developed for single cells, has spatial versions like stereo-CI-seq. These use antibodies tagged with DNA barcodes alongside RNA sequencing.
So you capture RNA and protein information simultaneously from the same location.
Exactly. You get gene expression and protein abundance measured together, spatially resolved. Similarly, methods are combining spatial ATAC-seq with RNA-seq, or spatial CT and tag with RNA-seq to link the epigenetic state directly to gene expression in the same region or cell. This gives you a much more integrated, holistic view of cellular function.
That's just astounding, seeing all these layers at once. Okay, we've covered the where in incredible detail, but biology doesn't stand still. How do researchers add the crucial dimension of time into these spatial maps?
Yeah, that's the next frontier. Making these maps dynamic, turning the Google Maps into a Google Earth time-lapse, maybe? Most spatial omics studies currently provide a snapshot at a single time point.
A static picture.
Right. But dynamic processes like development, regeneration, or disease progression happen over time. So integrating temporal information is critical. The main approaches involve using DNA barcodes as a sort of molecular clock or lineage tracer within the cells.
How does that work? Like a cell writing its own history?
Sort of. Techniques like IntMemoir use engineered genetic systems where enzymes, like integrases, make cumulative changes, basically edits or scars, to a synthetic DNA barcode sequence within the cell over time.
So the barcode sequence changes as time passes.
Exactly. By sequencing or imaging these evolving barcodes at different times, along with spatial omics, you can reconstruct the history of cellular events and trace cell lineages through space and time. Other methods like space-TREX use viral vectors to introduce unique genetic barcodes that are passed down to daughter cells, Combining this with spatial transcriptomics lets you map out clonal relationships and track cell fate decisions within the tissue structure over time.
So you can see not just where cells are, but where they came from and what they're becoming, all within the tissue context.
That's the idea. It's still challenging. Things like the efficiency of barcode integration and the accuracy of reading them out over time are hurdles, but it's a really exciting direction for capturing biological dynamics.
Absolutely. Okay, so researchers are generating these incredibly rich multidimensional data sets, RNA, protein, epigenetics, location, maybe even time. This must be a monumental amount of data. How on earth do they process it all and actually make sense of it?
You're right, the data analysis is a huge challenge and a field in itself. It goes way beyond just listing molecules. You need sophisticated computational tools. The basic workflow usually starts with pre-processing, cleaning up the data, filtering noise, normalizing signals.
Standard stuff.
Right. Then, depending on the resolution, you might segment the tissue image into regions or identify individual cells. A key step is often annotating cell types, figuring out if a spot or segmented cell is a neuron, an immune cell, a cancer cell, etc., usually by looking at its molecular signature.
Okay, then what?
Then the deeper analysis begins. Researchers look for spatially variable genes, SVGs, genes, whose expression levels systematically change across different locations in the tissue. These SVGs are often critical for defining functional domains or boundaries.
Like genes only turned on at the edge of a structure.
Exactly. They also try to infer gene regulatory networks, GRNs, to see how genes work together in specific spatial contexts. And for data that isn't single-cell resolution, they use deconvolution algorithms to estimate the mix of cell types present in each measured spot.
That sounds complex.
It is. There are challenges like data sparsity.
Mm-hmm.
You don't detect every single molecule background noise and dealing with densely packed cells. But the tools are getting better fast. Another big area is analyzing cell-cell interactions. By looking at which cells are neighbors and what signaling molecules, ligands and receptors they express, researchers can map out communication networks within the tissue.
Seeing who's talking to whom chemically.
Precisely. And since tissues are 3D, a lot of effort goes into 3D reconstruction. Tools like PACY and Spatial can take multiple 2D spatial omics slices and computationally align them to build a 3D model of the tissue's molecular architecture.
Creating the full 3D map.
Yeah. And when you add the temporal data we talked about, you get into spatiotemporal trajectory analysis, trying to trace how cells move, change state, and differentiate over time within that 3D space. It's computationally intensive, but incredibly powerful.
And where does all this data live? There must be databases springing up.
Absolutely. The sheer volume of data necessitates large, centralized repositories. Major initiatives like the Human Cell Atlas, HCA, and the Human Biomolecular Atlas program, HIBMA-MR, are coordinating efforts to generate and house single-cell and spatial data for various human tissues. There are also more specialized databases emerging just for spatial transcriptomics data, like StomachDB or SpatialDB, and for specific areas like cancer, HTN, or neuroscience, Allen Brain Map.
What are the challenges for these databases?
Key challenges include handling the multimodal nature of the data RNA, protein, imaging data all together, ensuring really good metadata so people know exactly how the data was generated, making the interfaces user-friendly, and just having the computational infrastructure to store and process everything at scale. Integration is key.
Okay, this technology is clearly cutting edge and generating incredible biological insights. But let's talk about the real-world impact. How is spatiotemporal omics actually being applied today to understand life and potentially improve human health?
The applications are already incredibly broad and growing fast. Because it captures that spatial context, it gives us insights we simply couldn't get before about how cells interact locally to create functional tissues.
So, like understanding organs better.
Definitely. That's a major area. Elucidating organization and function. Spatial omics lets us build detailed maps of organs, identifying cell types, their densities, their interactions, and the molecular networks that define different functional regions.
Can you give an example?
Sure. A great example is the recent macaque brain atlas generated using StereoSec. They mapped over 40 million cells. This allowed them to identify hundreds of distinct cell subtypes and precisely map where they reside in different brain regions, revealing organizational principles of the primate brain at unprecedented resolution. Similar studies are mapping the liver, showing gradients of gene expression across functional zones, the heart, finding rare but crucial cell types, the lung, the testes. Even in plants, mapping differentiation in leaves.
Wow, creating these incredibly detailed atlases. What's needed next there?
moving towards more multi-omics atlases, integrating epigenetics and proteomics spatially, and developing standards for building true 3D atlases that let you visualize the whole organ context are key next steps.
Okay, besides mapping existing organs, what about the ultimate biological process? Development. How a single cell becomes an organism.
That's another huge area where spatial omics is transformative, unveiling spatiotemporal dynamics during development. It's allowing us to dissect how organs form moment by moment, molecule by molecule.
Oh, so?
We can now track how different cell populations arise, migrate, and interact to build complex structures like the brain, heart, or gut during embryonic development. Studies in humans, mice, zebrafish, flies are revealing intricate details about organogenesis, how specific regions get patterned, how different cell types specialize. For example, seeing how specific types of neurons migrate to their final positions in the developing brain.
And what about regeneration, when tissues repair themselves?
Yes, it's also shedding light on tissue regeneration. Studies in animals like axolotls, which can regenerate brain parts, are using spatial omics to find stem cell populations that resemble early developmental cells. It helps us understand the molecular programs that drive repair in the liver or regeneration in organisms like planarians.
It sounds like we're watching the instruction manual of life unfold in space and time. What about the bigger picture evolution?
That's a fascinating angle, understanding evolution at spatiotemporal resolution. Genomics tells us about genetic changes between species, but spatial omics helps connect those genetic changes, genotype, to the resulting physical traits, phenotype. By comparing spatial gene expression maps across different species, we can see how changes in gene sequences lead to changes in where and when genes are turned on, which in turn affects cell types, tissue structures, and ultimately organismal form and function. For instance, researchers have used it to identify potentially human-specific cell types in the brain by comparing human brain maps to those of other primates. It's also being used to study how complex structures, like parts of the cerebellum, evolved through duplication and divergence of cell types, or how plant leaves adapted for different types of photosynthesis.
That's a really powerful way to connect genetics to function across evolutionary time. Okay, let's turn to perhaps the most anticipated application, disease. How is this technology changing how we understand and potentially combat diseases?
This is where the potential for clinical impact is enormous. While large genetic studies link variants to disease risk, spatial omics can show us the exact impact of those variants on specific cells within the affected tissue. It pinpoints the cellular and microenvironmental changes that actually drive the disease process.
Getting much more specific than just knowing someone has a risk gene?
Exactly. Take cancer. This is a huge area for spatial omics. It allows researchers to map the incredible heterogeneity within a tumor, seeing different subclones with different mutations, mapping areas of hypoxia or inflammation, and crucially, studying the tumor microenvironment, TME.
The ecosystem around the tumor cells.
Right. How cancer cells interact with immune cells, fibroblasts, blood vessels right there in the tumor. This is critical for understanding why some tumors resist treatment or why immunotherapies work for some patients but not others. For example, seeing how specific types of cells called cancer-associated fibroblasts might block immune cells from attacking the tumor, or identifying structures like tertiary lymphoid structures within the tumor that correlate with better response to immunotherapy.
So it could guide treatment choices.
That's the hope. It's also being applied to chronic and infectious diseases. In Alzheimer's, it's helping map the interactions between microglia and astrocytes around amyloid plaques. In COVID-19 research, spatial methods helped show how SARS-CoV-2 might infect specific support cells in the nose, potentially explaining the loss of smell.
It provides such a detailed, localized view of the disease process. The potential seems massive. So what's the path forward? What will it take for spatiotemporal omics to move from the research bench fully into the clinic to actually change patient care?
That's the key transition. It's definitely foundational for precision medicine. By decoding the tumor microenvironment or understanding disease progression at this resolution, it offers huge potential for better diagnostics and identifying personalized treatment strategies. It's already playing a role in identifying better biomarkers, especially for guiding complex treatments like immunotherapies.
But what are the hurdles for routine clinical use?
Several major ones right now. Cost is still high. The turnaround time for generating and analyzing the data can be long. There could be inconsistencies between different platforms and methods. And the bioinformatics needed to analyze the complex data requires specialized expertise, which isn't always available in a clinical setting.
How might we overcome those?
One very promising direction is integrating artificial intelligence, AI. The idea is to train AI algorithms on vast amounts of spatial omics data, paired with standard histology images, the H&E stained slides that pathologists look at every day.
So the AI learns to predict the molecular details just from the standard image.
Exactly. If AI can reliably infer gene expression patterns or cell types directly from routine H&E stains, it could drastically reduce the cost and time associated with direct spatial omics testing, making the insights more accessible. But this requires rigorous validation, standardized protocols, and large clinical trials.
That could be a game changer. What about diagnostics for tricky cases?
Spatial multiplexing, analyzing many gene or protein markers spatially, could be huge for diagnosing complex diseases, like tumors that are hard to classify using traditional methods. Instead of just one or two markers, you could look at dozens plus their spatial relationships on a single slide. Techniques like stereo C8-ESEC could potentially run standard diagnostic panels and add new predictive biomarkers simultaneously.
So what's the checklist for getting this into routine practice?
We need standardized, robust, maybe even automated protocols. The results need to be easily interpretable and reproducible. It needs to integrate smoothly into existing digital pathology workflows. Costs need to come down significantly. And ideally, it should require less specialized expertise to run and interpret, making it more accessible beyond major research centers. Ethical and legal aspects also need careful consideration.
It's a big list, but the potential payoff is huge. Given the scale of this technology and the data it produces, are there big collaborative efforts driving this forward globally?
Oh, absolutely. Large-scale collaborative science is really the way to go for something this complex, building on the model of the Human Genome Project. We've already mentioned the Human Cell Atlas, HCA, and HubMap. These are massive international efforts to map all human cell types and tissues, increasingly incorporating spatial data. There's also the Spatiotemporal Omics Consortium, STOC, specifically focused on using these methods to answer biological questions over time, and dedicated efforts in specific fields like the Brain Initiative Cell Census Network, BICCN, generating incredible brain atlases, and the Human Tumor Atlas Network, HTN, focused on mapping cancer progression spatially and temporally.
What are the main challenges for these big collaborations?
Coordination is always key, ensuring efficient teamwork across many labs and disciplines. Fostering
So wrapping things up, we've really explored how spatiotemporal omics is giving us this unprecedented Google Maps of the body. It's not just a static map, but one that can incorporate different molecular layers and even the dimension of time. It's an incredibly powerful lens on life's complexity.
Absolutely. This convergence of spatial technologies with sequencing advances, single-cell analysis, and AI is fundamentally changing how we study biology. And it holds immense promise for transforming how we diagnose, treat, and ultimately predict disease, really accelerating that goal of true precision medicine.
Thinking about this incredibly detailed, dynamic map of the human body we're starting to build, what specific biological puzzle or medical mystery do you think spatiotemporal omics might crack open next? And maybe looking just a few years down the road, how do you see this changing the practical reality of diagnosing and treating patients? What should we be watching for?